This is the pre-recording, the prep for lecture 21, and we're going to be looking at, so the first lecture was really about sort of gene exchange in bacteria, the different ways that happens. Oh, great noise. Hopefully temporary. I sort of introduced the idea of interrupted mating, and we will be doing that in this lecture. So the idea is you get a rough idea of the relative order of genes and approximately how far apart they are. For co-transformation, that is just a way of finding out whether two genes are close together within a certain distance on the chromosome or far apart from each other. That's probably a good starting point. And then we're just going to start talking about HFR, high frequency recombination mapping. This is very precise and detailed, requires careful design so that you select exactly what you think you're selecting. We will just talk about replica plating as a way of figuring out the genotype of all the colonies, because you'll make hundreds, thousands of colonies in an HFR map, and each colony has to be genotyped separately. Okay, so this is, oh, I didn't take off the pen. Okay, so basically when we map genes by interrupted mating, we take an HFR strain of known genotype and an F- strain of known genotype. And we have a set of genes, let me change the color here, a set of genes that we plan to map. So we're going to map a bunch of genes, right? But then we also need a selective marker. So we want to have on the HFR strain something like a sensitivity to an antibiotic, so that, you know, we're going to mix HFR and F- cells together. The HFR strain after the mating process is over is still intact. There's nothing kind of missing. And so those cells will be in the culture, and we don't want to count those because we'll think, if we count them and figure out the genotype, we'll think that all of these genes have been incorporated into the recipient cells, which will not be accurate. And so we need a way of making sure that the HFR cells are all removed from the culture that we're going to test. We also, the way we do that is we use an F- strain that is resistant to that antibiotic. So we use strep resistant, say, for this example, and strep sensitive HFR. And this gene has to be far away from the ones that we're actually mapping because we don't want it to accidentally be incorporated, right? This is where all our figuring out is going to be, and the other genes here are for selection. Now the F- cells, there may be some cells in the culture that haven't participated in mating. And if they haven't, they're going to have all of these genes. And so, you know, these are auxotrophic alleles of the genes in question. And we don't want to be counting these in our total either. How we decide on the total that we divide by when we're trying to figure out the recombination frequency between two genes is very important. We have to have the right kind of total. And so what we want is we want to have cells where at least mating was initiated. And so we're going to have prototrophic alleles of genes, like they can make their own amino acids or biotin or whatever it is, on the HFR strain. And the F- strain cannot make those themselves. So then we allow the mating to take place, we interrupt it, I'll talk about that in a minute. And for each aliquot, we plate them on strep to get rid of these cells, and we plate them on media that lacks these nutrients, whatever they are. So the F- cells and the two parental types are missing. And then what we have is we have a bunch of F- cells that could have incorporated the wild types for the genes in question here. And then we do an experiment to see, you know, does the D gene come in to the recipient cell before the B gene, and so on? What is the relative order and the approximate distances between them? Okay, and so this is the one we're actually going to work with. So we have our strain A, which is our HFR strain, and our strain B, which is the F minus strain. These are the ones we use for selection against the F minus cells because they can't make their own leucine and threonine, but the HFR cells can. And then this is the one we use to select against the HFR strain. And then the genes that we're going to map, they're shown in the middle here, but there's a lot of space between this region and the area that we're mapping. Okay. And this is just a description of the things I've just said. So threonine and leucine, streptomycin is an antibiotic, so cells can be either sensitive or resistant to it. Threonine and leucine minus, they cannot make their own of these amino acids, so they have to be provided in the media. Sodium azide is a toxic substance that bacterial cells can be sensitive to or resistant to, and that means they can grow on the toxin. They have a biochemical way of inactivating the poison. Bacteriophage T1, this stands for T1, but without the E. You can be sensitive to it, susceptible to being attacked by that virus, or you can be resistant to it. It's a difference in the proteins on your protein coat on the outside. There are these cell surface proteins that the virus recognizes and attaches to in order to attack the cell. And if you don't have those proteins on your surface, then you are resistant to that virus. And then the LAC and the GAL genes are lactose and galactose. These are sugars. If you can use them, you are LAC plus or GAL plus. And if you cannot digest them, then you have minus. So if we plate these only on those sugars, then if they're minus, they're going to die because they don't have any sugar source, any carbon source. Okay, so those are what the genotypes are, and here's our experiment. And so it was already known, so this is information that was obtained previous to this mapping experiment using interrupted mating. And it was known that threonine and leucine genes had entered early from this particular strain. And then to map these other genes, you basically mix the strains in liquid culture at time 0, and then every 1 to 2 minutes, probably they wouldn't start until about 5 minutes, but every 1 to 2 minutes they would take a small sample of that and they would interrupt the mating process by putting the cells into a blender and turning the blender on. Bacteria stop mating really fast when you do that. It sort of breaks that little tube, the connection between the cells. And then you take those cells and you plate them on media with streptomycin to select against strain A, and that has everything added to it except threonine and leucine that selects against strain B. Okay, and then you've got a whole bunch of recombinant cells, and now you want to figure out what their genotypes are for each of the aliquots at all of these genes. Because at this point, we have information about this and this, but we don't know the order or the relative distances between those genes. That's the information we're trying to find out with this experiment. Okay, so you've grown those cells in the medium with no leucine or threonine, but with streptomycin. So that basically selects for this, and the only way you can get those together is if there's been some sort of crossing over between the two chromosomes. So we're selecting cells where we know some crossing over has taken place, and then it's just which other genes have been incorporated, right? So this is just an example. So this is our original plating experiment. We plate a bunch of cells after 2, after 4, after 6, after 8, 10, etc. Minutes, and whatever cells grow on these colonies, suppose there were some, 2 minutes is probably too early, but you know there's going to be a certain number of colonies on those cells, and then we take those and we do replica plating, which I'll show you later exactly how it works, and we plate the colonies on full medium plus sodium azide. And if the colony grows, then that means it's azide resistant. We plate it on a cell media that also has phage added to it. There's a special technique you use for that. And if they're susceptible to that phage, then they will, it's like a cleared area. You can see which cells got infected. And they form a particular structure that you can observe. And then you give them only galactose as a sugar source if they grow their Gal plus. Same with the lactose if they grow their Lac plus. And so you can take the same batch of colonies and plate them on a bunch of different media and see what the genotypes are. And it requires the information from all of these to get the genotype of each individual colony. So it's a lot of work. It's a lot of labeling, careful orientation of the plate, knowing which colony was which on the original plate and which, where those are located on those plates. But imagine, that you've got 50 colonies or 20 colonies or something. You have to follow each colony and where it is on the plate. So it's like an exercise in record keeping and careful attention to detail. And so basically, if you take a look at these data, this kind of tells you how the experiment went. So, say they were even every 1 minute or so, they were taking another aliquot and testing it. They didn't start to see any cells that were resistant to sodium azide until approximately 8 minutes. So in around this time period, they saw a very small number of sodium azide resistant cells. And then at about 10 minutes here, you got the T1 resistant cells. And then not until about 17 minutes had passed did you start to detect some Lac plus cells. And then not until 25 minutes were you able to start recognizing some Gal plus cells. So if you're going to draw a map of this, you would have the little arrowhead that indicates where, the beginning of transfer. And we already know that Lu and 3 are somewhere along here. And we weren't monitoring them particularly because we were using them as our selection. But about 8 minutes afterwards, we start to see the sodium azide resistant cells, right? And then 2 minutes later, the T1 resistant cells. So we know there's about 2 minutes between here. And then the Lac plus is a little further along, L plus, and that was 7 minutes later, right? And then Gal plus about 8 minutes later. And so notice the units are minutes, not map units, and they don't really, because some the rate of transfer isn't necessarily 100% consistent all the time. It could slow down a little bit and speed up a little bit. This isn't super accurate, but what it does tell you is that we know that this gene enters first, and we know that it's relatively close to this gene. And that these two genes are, more than twice as far apart as these two, and that this one is quite distant, right? There's what, 25, so there's 17 minutes difference between this gene and this gene. So we know the relative order, and we can tell which ones are close and which ones are far apart based on this kind of information. Oh, I forgot that I put that. That's my little animated version. And then here's a nicer one, but it shows you approximately where things are located, and it also shows you how to make the map. So you would put the arrow here to indicate that this is the direction of transfer starting here, and then you'd put the map distances along here. And then I wouldn't suggest necessarily a little box here. I would just have a line, because we don't actually know the relative sizes of these. So you'd be better off with just kind of having a line. I would suggest it that way. Okay. And so, this is a really small portion of the whole bacterial chromosome. So if we were looking at, the entire chromosome, we're probably only mapping like this little tiny portion of it. And so to get a really good picture of the bacterial chromosome, we need to do lots of different overlapping experiments. And again, I'll just remind you what I just said. The units in this case are minutes, and minutes are not strongly equivalent to map. They're roughly related to map units, but we would not consider this particularly accurate. So we wouldn't go, oh, you know, there's 8 minutes between here and here, so maybe that's equivalent to 8 map units. No, it's not equivalent to 8 map units, and it's not equivalent to 4 map units particularly either. So you just want to make a note between these. These are not useless. They tell us some information. Genes that are close together, genes that are far apart, and their relative order, that's all perfectly accurate. But the exact distances are not accurate. So these interrupted mating experiments, I've just said this, they tell you the relative order and the approximate distances of genes, and in relatively small sections of a chromosome at a time. So we only study a few genes at a time. But the good news is that if you look in E. coli, there are lots of places all over the bacterial chromosome where the F factor can actually insert into those bacterial chromosomes. So there are some strains where the F factor, you know, has inserted here in this orientation. There's other strains where it's inserted in approximately the same place but in the opposite orientation. You know, there's many different positions and orientations, so that we can do an experiment, you know, with these two, and an experiment with this one, and this one, and this one, and so on. And when you combine the information we get from these different experiments, we get a pretty reasonable picture of what the bacterial chromosome looks like. And we actually use this kind of information as a way of designing our experiments for doing HFR mapping, which I'm going to talk about in the next lecture, to get those really precise and really accurate information. Knowing the relative order in advance and of the genes that we're trying to map, which one enters last of the other two, then that really helps us with our design of our experiments to get a really precise map. Okay, I'm going to stop here because I want to take the time to go through it, and I don't want the video to be too long. So we'll start here again on the next prep.